Was there with you guys today, but I really appreciate the chance to share some of our data. So as Cheryl said, our group works on T-cell responses to tuberculosis, and we use mouse and non-human primate models. And today I'd like to show you some of our more recent data. We're trying to develop strategies to manipulate T-cell function to treat TB. And first, I'm going to show you some of our work on PD-1, which still continues to this day, on it and its role in MTB infection. And then secondly, I'll show you some more newer work from our group where we're trying to branch out into unconventional T-cell populations, such as mucosal-associated invariant T-cells or mate cells, and asking if one can target this kind of population of T-cell for the treatment of MTB infection. So if not for COVID, I'd be telling you that tuberculosis is the leading cause of death due to a single infectious disease, but I'm sure this time next year or the year after, I'll be able to tell you that tuberculosis is the leading cause of death due to a single infectious disease. And there really is a huge need for the development of new prophylactics and therapeutics for the treatment of MTB infection. I think first and foremost, there is a huge need for the development of effective vaccines. And this is where I'd say that the lion's share of the work in TB is really taking place. The only vaccine we have for TB is 100 years old. And while it does prevent severe forms of TB in infants, it doesn't do a whole lot to protect adolescents and adults from tuberculosis. And that's where all the transmission happens. And so there really is a huge need for the development of new effective vaccination regimens in TB. And that would probably have the biggest impact on the global burden of tuberculosis. There's also a huge need for development of novel antibiotic therapies, even though most cases of TB are actually due to drug susceptible strains of TB and can be clinically cured. It takes 6 months. And there's the ever-present threat of drug-resistant strains of TB. And so new chemotherapeutic regimens, which can dramatically shorten the treatment, are really desperately needed. And we're learning more and more now also that clinical cure often does not mean microbiological sterilization of the infection. And so we really do need more effective antibiotics that can achieve a sterilizing cure much more rapidly than the 6 months that it takes now. Lastly, a topic which I think gets a lot less attention, but is something that is very near and dear to our heart, is the concept of host-directed therapies, which is sort of the catch-all term for immunological interventions. The idea is not, instead of giving a chemical which will kill the bacteria, but try to manipulate the host immune response to convince it to kill the bacteria, or to limit the detrimental pathology that happens in the lung tissue as a result of MTB infection. Now, one of the key interactions that determines the outcome of tuberculosis is the CD4 T-cell macrophage interaction. TB grows inside of macrophages, and control of TB really requires that a CD4 T-cell nestle up against a TB-infected macrophage and provide signals to that macrophage that tell it to restrict growth of TB, or if we're lucky, even kill a few bacilli. Now, even though without CD4s, the infection is never controlled well. CD4s, most of the time, we're unable to actually eradicate the infection. And so a major question we have is, can we actually manipulate T-cell function in a way to enhance control of MTB? And I feel like I've been asking the same question about PD-1 and its control of chronic infections for over 15 years now, but we're still asking, what is the role of PD-1 in chronic infections in our group? And so, of course, I think it always feels weird to talk about PD-1 to a group of folks at Emory, but just to remind any of the new students, perhaps, PD-1 is an inhibitory receptor that's expressed on the surface of activated T cells. And when it's engaged by one of its ligands, it blunts T cell receptor signal transduction as well as CD28 dependent co-stimulation. And so if you block the interaction between PD-1 and one of its ligands, you boost the function of the T cell. And this is particularly important in the context of persistent viral infections and perhaps most important in cancer. In fact, there's a number of drugs now that are targeting the PD-1 pathway in cancer, and the Nobel Prize was awarded to Jim Allison and Tasuku Hanjo for the development of PD-1 targeting reagents for the treatment of cancer. And so basically, it feels like every other infectious disease field is asking themselves, can we do this too? And this in the TB field is no different. There's been a long-standing interest for several years now, based on these successes in cancer in the TB field, saying, well, that should all work in TB too as well, right? And here's an example of a cartoon from a review article I lifted where they actually directly draw a parallel between a tumor cell and a TB-infected macrophage and blocking the PD-1 pathway to enhance control of the infection. And so this has been an outstanding question for quite some time. Now, the first data on the role of the PD-1 pathway in the context of MTB infection actually came from the mouse model, first from Bill Jacobs' lab, and then soon after by Alan Schur and I, where we actually found that rather than be more resistant to TB infection, as you might expect when you remove an inhibitor, the mice are actually extremely susceptible to MTB infection. In fact, they die just as fast as about any other immune gene knockout mouse we've ever seen. And this is the only, or the first example, I should say, of an inhibitory pathway leading to susceptibility. Normally, when a knockout mouse dies of TB, it was a mouse that lacked a pro-inflammatory axis, like CD4 T, interferon gamma, IL-1, TNF alpha, or CD4 T cells altogether. This was removing an inhibitory pathway and seeing a rapid death of the animal. And we were able to actually show that this was the CD4 T cells themselves that were leading to the early death of the mouse. So when you take away PD-1, CD4 T cells, and a little bit of a contribution CD8 T cells, they wind up killing the mouse instead of controlling the infection. And we don't know exactly why that is, but we were able to go on to show that at least it has something to do with interferon gamma. As I mentioned in the last slide, interferon gamma is one of the sort of archetypal, most important cytokines in host defense against TB. And so you might expect that if you boost T cell function, they would make more interferon gamma and that would enhance control of TB. But instead, it leads to a huge amount of necrosis in the lung tissue. And then TB grows in these necrotic regions of the lung and reaches really high levels and kills the mouse. So what we know based on the mouse model data is that PD-1 seems to actually be playing a host protective role because it's limiting detrimental inflammation. So the problem is that that's just the mouse data, and mice are not really the best model for tuberculosis. However, I also told you that there's a whole bunch of people being treated every day with PD-1 blocking agents as cancer immunotherapy. So you might think if PD-1 was so important in host resistance to TB, you'd see people getting tuberculosis. Now, it turns out that actually is the case. Over the past 4 or 5 years, there's been a steady output of case reports of people developing tuberculosis after receiving PD-1 blocking drugs for cancer. In fact, the product label of atezolizumab actually has tuberculosis on it. There were cases of TB visualized. So this actually seems to jibe with the mouse data. That maybe PD-1 is actually a host, this inhibitory axis is actually an important part of host resistance to TB, which is a somewhat counterintuitive on its surface. The problem again is that, as I said, the mouse data is mouse data, not the best model for tuberculosis, even though they are very powerful animal models. And the problem with the human data is TB is kind of everywhere anyway, and these are all just case reports, and these individuals they had cancer, they had other things they were being treated with. So it's really not clear based on this mouse and human data if there really is a role for PD-1 in host resistance to TB. So we thought, well, this is probably worth just testing in rhesus macaques because they really are the gold standard model for tuberculosis, the animal model of tuberculosis, because they pathologically recapitulate all of the different cardinal features of tuberculosis that you'd see in a human, and the immune cell populations of macaques and the molecules they express and the quality of T cell responses way more similar between macaques and humans as compared to a mouse and humans. So we set up an experiment where we infected 12 macaques, 6 got ice-type control, and 6 got a rhesus macaque primatized anti-PD-1. This was Gordon Freeman's clone. So we picked the key thing about the study is that the timing and the strain and dose of TB. We were hypothesizing that PD-1 blockade would make TB worse. And so we picked a dose and strain of TB where we'd have slow progressive disease in the control animals so that we could see a deflection of a worse disease. We can basically have any outcome of tuberculosis in a macaque, and we thought if we go too virulent, we'll mask the immunopathology that we're predicting to see with the virulence of the bug. So we went a little bit low on the virulence of this outcome. And we started the treatment right around 2 weeks post-infection, which is right about the time T cells first starting getting primed in the draining lymph nodes of the lung. So this isn't a therapy. We didn't wait until the animals were chronically infected and started to develop TB and then tried to block it. We were actually asking, what is the role of the PD-1 pathway in TB infection throughout the course? So this is more of a trying to mimic what we'd see in a knockout mouse rather than what might happen in a cancer patient. Now, we just started by looking at a few gross clinical readouts. As I mentioned, all of the control animals were healthy. They continued to gain weight and had no overt signs of tuberculosis throughout the course of the disease. And in fact, you could probably have diagnosed them as latent at this time point. In the anti-PD-1 treated animals, we had one animal develop acute signs of distress and need to be euthanized early from a collapsed lung. And we had another animal develop cachexia, but make it to the endpoint. Now, we also longitudinally followed disease progression in these macaques using FDG PET-CT imaging. So this allows us to monitor inflammation in the lungs longitudinally over time. There's no great way to measure bacterial burden in the lungs of an animal. You have to euthanize it, carve out the lungs. And so in macaques, which are outbred, you really just can't sacrifice different groups like you would with a mouse experiment. So this gives us a great power to actually longitudinally progress, follow disease. And so you can see disease was pretty stable after week 4 when you first start to get a PET-CT signal in the controls. But we had several animals where disease was progressive after PD-1 blockade. Interestingly, this one animal here, which actually had the worst PET disease progression by PET, actually had continued to gain weight throughout the study. So just collectively with this simple weight loss, PET-CT, and survival, none of the control animals had any signs of disease. But 3 of the 6 animals seemed to have something go wrong. One had to be euthanized from severe distress. One became cachectic, and another had rapidly progressive disease by PET-CT imaging. But really what matters is what happened to the bacterial burdens. So the way we measure the immune response in the bacterial growth in the lungs is at necropsy, we carve out the whole lung and we bread loaf it. And we actually comb through centimeter by centimeter the entire lung bed, and we carve out every palpable granuloma and we process them individually. So every single dot on this graph is a granuloma that we pulled out from the animal. So all of the analysis I'm going to show you is at the individual lesion level. And what you can see, this is sort of the punchline of the whole first part of the talk here, is that bacterial loads were increased after PD-1 blockade. So we didn't enhance control of TB for sure. And we quite clearly actually increased bacterial growth when we blocked PD-1. Now, when we break it down by individual animals, it was 4 out of 6 that had an increase in bacterial load. And of course, there's a huge spread that's expected in macaques, but it was a pretty significant increase in bacterial loads. So given that we actually made TB worse, the question was, well, what did we perturb about the immune response when we blocked PD-1? Now, I'm going to show you a number of different analyses we did that were all based on specific hypotheses we had about the role of PD-1, but I can't tell you exactly why the bacterial loads went up. I can just tell you what changed as along with this increase in bacterial loads. We don't know mechanistically exactly why this happened. The first thing we looked at was the kinetics of the tuberculosis specific T cell response in the airways, because we can longitudinally sample the airways and look for the accumulation of the TB specific T cells in the lungs. And we do that by restimulating with these megapools of peptides derived from TB from Cecilia and Alex. And we can track the kinetics. And surprisingly, there was we expected that we'd see this huge increase in the kinetics of the T cell response when we blocked this inhibitory receptor, but that's not what we saw. We actually slowed down the CD4 T cell response in the airways after PD-1 blockade. Here, eventually they were pretty close by the end, but certainly it seems as if we delayed the accumulation of T cells in the lungs. And that was one of the first things that really diverged from mice that we did not expect. We had the same kind of clinical outcome and microbiological outcome, but we didn't see this huge increase in the magnitude of the T cell response like we expected, at least not in the bowel. Now, when we looked in the individual granulomas, again, each dot is a granuloma that we restimulated with peptides and did intracellular cytokine staining on to quantify the percentage of cells which are specific for TB. And when we looked at the CD4 T cells, there was no difference. So we didn't actually boost the magnitude of the T cell response, the CD4 response at all, but we did for the CD8s. So if you look at the CD8 response here on the right, there's a significant increase in the frequency of tuberculosis specific CD8s. We also looked for many different cytokines, IL-2, IL-17, CD153, a number of different molecules of interest in the lab. And there were no difference in the functional capacity of the CD4 T cells. So we didn't make more of them and they weren't more functional. But as we were phenotyping the CD4 T cells, one thing that really stood out was after PD-1 blockade, the CD4 T cells in the granulomas dramatically upregulated CTLA-4, which is a different co-inhibitory receptor. So by blocking PD-1, we didn't boost CD4 function. If anything, we slowed them down in the lungs. and they upregulated a different inhibitory receptor. When we look at the CD8 T cells, they actually did look more functional. And one of the biggest differences we saw there was a significant upregulation of granzyme B, which is a marker that CD8 T cells use to kill target cells. So we don't know which T cell may have been driving the increased bacterial loads, but we had a much bigger impact on the CD8 T cells than we did the 4s, which was different than the mice where the CD4s were primarily impacted. Now, we also wanted to look at the quality of inflammation in the granulomas. So we ran a cytokine multiplex assay, just a classic Luminex. Again, each one of these dots is an individual granuloma where we measure the amount of soluble cytokine by Luminex in that granuloma. In the top 3 cytokines that were increased were interferon gamma, IL-18, and TNF. So the Th1 associated cytokines, which is exactly what we saw in the mice. Those were the most significantly increased. Now, we sent the data to a buddy, Bruno Andrade at Fiocruz in Salvador, who's a statistical master. And he did something that I'm going to butcher the explanation on, but a canonical correlation analysis, which basically is a network-like analysis that looks to see how all of the data points are interconnected with one another and can graphically represent the quality of inflammation based on how those networks are associated amongst the different groups. And what you can see is you can very clearly distinguish the pattern of inflammation in the PD-1 versus ice-type control treated animals. And when you looked at which of the factors which were driving this difference in the pattern of inflammation, IL-18 really not only was increased, but associated with driving this big difference. Bruno also went on to ask how each of these different analytes correlated with bacterial loads. So here I'm showing you the red bars are the statistically significant positive associations, in the blue, the negatives. What you can see here is in the anti-PD-1 treated macaques, there's just a lot more red bars. So many more inflammatory mediators correlated with bacterial loads, positively correlated with bacterial loads when we block PD-1. In other words, more inflammation, more bacteria. Now, one of the things that really stood out to us was this molecule interleukin-1 beta. If you look here in the control, there was no correlation between IL-1 beta levels and bacterial burdens, but after PD-1, it popped up to the top of the list. And that was interesting because that gave us an inflammasome signal, because of course, IL-18 and interleukin-1 beta are both cytokines which are produced in a proform, which must be matured by the caspase-1 inflammasome to mediate biological activity. So we thought we'd look to see if the inflammasome was being activated in these granulomas. And to do that, it turned out the best way was to look by Western blot. So we ran a crap load of Western blots on granuloma lysates, and we could see pro-caspase-1 in every gran. But interestingly, it was kind of hit or miss as to whether or not we observed that caspase-1 was activated. You look for activated caspase-1 by the presence of this P10 band down here. Now, when we looked at just what percentage of granulomas had an active P10 band or not, it was really not different, 11 out of 23 or 14 out of 23. So PD-1 blockade did not increase the likelihood of detecting activated inflammasome. However, when we looked at how that correlated with bacterial burdens as to whether or not the granulomas had an active P10 band or not, what we found is that all of the increased bacterial replication we found in the anti-PD-1 treated animals came from granulomas that had an active P10 band. So we hypothesized that maybe even though PD-1 blockade isn't boosting the inflammasome, if the inflammasome is active, PD-1 blockade makes things worse. I hope that makes sense. So to really kind of test that, we decided to go back to mice. So this will be the only mouse experiment I'll show you for this first story. And we crossed the PD-1 knockout mice to caspase-111 knockout, double knockouts and made triple knockouts. And what you can see is PD-1 knockout mice normally die very rapidly, but if you knock out their inflammasomes, that you can significantly extend their survival. So I think this is a novel part of a novel new mechanism associated with PD-1 dependent exacerbation of tuberculosis. We don't quite really understand it, but there's some interplay. If the inflammasome is activated in the granuloma, when you block PD-1, it's way more likely to increase bacterial replication. Now, we don't know if that's IL-1 or IL-18 or necrosis, the pyroptosis that's associated with inflammasome activation, but we need to dig into that a little bit further. Okay. Now, another hypothesis we had was that perhaps we'd be screwing up the movement of T cells inside of the granuloma. Now, there had been previous reports from Jeff Bluestone's lab and Ron Germain's lab saying that when you block PD-1, the T cell gets a stronger stop signal. And so they grab a hold of the APC better. And so we wanted to ask, if we could be affecting T cell motility inside the granuloma. So we developed this assay to track T cell immune cell movement in the granuloma. Essentially what we've done is as soon as we necropsy the monkey, we embed the granuloma in agarose, cut it into really thick slabs, stain it with antibodies, and then put it under the microscope. And we get about 2.5 or 3 days worth of movement before there's too much crawl out to really think that the data is useful anymore. And there's a huge amount of heterogeneity from one granuloma to the next, and it's enormous. But this is a not too untypical of a movie where you can see there's many different patterns of movement. There's static T cells, rapidly moving T cells, static B cells. In general, the macrophage dense core is static, and lymphocytes are what moves. And then we also find these clusters of CD4 and B cells out here in the periphery, we call granuloma associated lymphoid tissue, or GRALT, where you often have dense T cell B cell interactions. Now, as I said, there's an enormous amount of variability from one granuloma to the next. We need to quantify this very carefully. We realized that if we basically plot for each one of those tracks, the mean speed versus its variation in speed, we get 3 populations of cells. We get cells that have a low mean and low variation that seem to have just been stopped. And we get population of cells here, which have a high mean speed and a low variability in speed. So we say these are steadily moving. And then we have this population of cells that falls up here, which is high variability and intermediate levels of average mean speed. So we say that they're stuttering. We can actually identify those tracks, put them back into a MARIS and observe. That's exactly the kind of behavior that you see when you actually watch those tracks move. And these 2 movies here are from the exact same monkey on the same day. So you can see one granuloma is all stopped and one is all steadily moving. And that's just the kind of variability that we saw on these granulomas. But we made way more movies than we should have. And what you can see is that overall, there was a bit of a trend, not striking, but there was a bit of a trend that after anti-PD-1 blockade, fewer of the granulomas had abundantly moving T cells. So we don't know that it was actually from the PD-1 blockade or the high bacterial loads or what, but there was a hint that maybe we screwed up intralesional motility of T cells by blocking PD-1. Okay, so just to summarize here, I think there's evidence now from both mice, humans, and non-human primates that this inhibitory axis is actually protective in the context of TB. And so TB may actually be a major adverse event of immune checkpoint blockade as cancer immunotherapy. We have no idea how prevalent it will be. It was 4 out of the 6 monkeys, but we haven't seen a ton of cases, maybe not as many as you might expect. So it's really not clear if this is going to be a big problem or really not so much a problem at all overall. We'll know more once more TB endemic countries start receiving a lot of PD-1 blockade for cancer. We think it may have to do with it preventing detrimental inflammation. You need a little bit of negative regulation to stop really bad inflammation in the lungs. However, I also want to point out, and perhaps this is a subtlety, but we had slower CD4 accumulation in the lungs. We didn't boost their numbers. They had more inhibitory receptors, and they didn't traffic around as well. It's almost like we slightly made the CD4s defective. So it's also possible that CD4 T cells might need a little bit of tonic regulation of their T cell receptor through this negative regulatory pathway to allow them to do what they're supposed to do. I hope that's not too much of a finery, but it changes the way I think about this quite a lot, because it means that you actually need negative regulation to do your normal job, not that it's just limiting you. Okay, I'm going to shift to the mate topic now. So if there are questions, it's a good break point.
Yeah, Dan. There's a question in the chat from Rabin. Rabin Turvansium. So he's asking, could you be directly affecting PD-1 signaling in macrophages themselves instead of T cells?
Absolutely, and there's data in the tumor literature that could be happening as well. I think we'd have to go back to the mouse model to try and test that. Here in the macaque model, we have no idea why. We focused our analysis on T cells, but we really don't know that they were the cause. We know in the mouse model, we can adoptively transfer PD-1 knockout T cells into a recipient mouse and kill the mouse. And that happens even if you mix wild type CD4 T cells together with PD-1 knockout. So we know you can recapitulate the rapid fatality of tuberculosis in mice by removing PD-1 selectively from T cells. But that doesn't mean it's also not also important on macrophages. We just are not a macrophage lab, so we haven't focused on as much, but very good question.
Yeah, great. Okay, well, we've got another question from Anna Enriquez. She's a PhD student. So any plans to see the effect on bacteria itself? So curious if the effect of higher load is just host driven or could be pathogen driven.
I have no idea. That's a great question. I think we need to find some good microbiologists to work with to do that, but that's a very fascinating question.
Okay, and from BJ, the questions are really coming in now. So what is the expression of PD-1 and CD4 T cells from the granuloma? Are they higher than any CD4 T cells? So maybe like CD4 T cells in the granuloma versus.
Yes, that's a great question. Tuberculosis specific CD4 T cells in the granuloma are basically all positive. It's a unimodal distribution and they're the highest in the granuloma. But PD-1 expression by MTB specific CD4 T cells is always kind of underwhelming as compared to what you see for like a LCMV clone 13 specific CD8. So they have PD-1, but it's nowhere near as sky high as you've seen in other models of full-blown exhaustion.
Okay. And then you probably know, so Rafiq Sekali is here also, he's in right now. So PD-1 is also expressed on an effector Tregs. So could you maybe comment, I guess, on perhaps whether more inflammation is triggered by PD-1 blockade on the Treg side?
Can't exclude that hypothesis at all. All I can say is that we looked at the frequency of Tregs and their a couple of activation markers, I think, and they were identical before with and without PD-1 blockade. But I can't tell you that that's not playing a role. It very well might be.
Yeah. A couple more questions. So John Altman has asked, so in the movie, why are B cells moving faster than the T cells?
It depends on the movie. They're not always.
Okay. And then, so Christina Niavi is also a PhD student now, well, a PhD student in Rafi's lab now in the IMP program. Did you look into ion channel regulation in terms of both macrophages and T cells? There are predominant ion channels in both populations that could be inflammasome related, maybe the role of T cell in NLRP3?
That's a fascinating question. We are following up on that role of the inflammasome with Catherine Meyer-Barber's lab in the mouse model at NIH. So hopefully I can tell you more later about that, but we never looked at that specifically.
Great. Okay, so I think that's the questions I see in the chat for now. So we'll let you go on, I think, to the next part of your talk.
Thanks, everybody. All right, and now for something completely different. So, we really have failed to target conventional T cells to boost the control of TB. If you vaccinate with peptides for conventional T cells, nothing happens. If you block PD-1, you kill the mouse. But there's a lot of interest in the field of other subsets of T cells, unconventional populations of T cells that bear invariant T cell receptors that could be really important. And in particular, I want to talk about our lab's interest in this cell type called MATES, or mucosal associated invariant T cells. Now, MATES are fascinating. Instead of having peptide-specific T cell receptors that are polyclonal, they have invariant T cell receptors that recognize a small molecule that's derived from the biosynthetic pathway of riboflavin synthesis in bacteria. And so it's actually even more complicated that there's an intermediate in riboflavin biosynthesis pathway, which reacts with an intermediate from glycolysis, which forms this extremely short-lived molecule called 5-OPRU. And that is actually then captured by an MHC class 1-like molecule called MR1. Now, MR1 is extremely non-polymorphic. It's one of the most conserved genes in the mammalian genome. And so this is a T cell pathway. This is a population of T cells which is present in mucosal barriers, which is sensing microbial metabolism, for lack of a better word. And they've been shown to play a role in a couple of different settings. But just the short of it is there's a number of different in vitro data and clinical correlations indicating that MATES may play a role in TB. There's been quite a bit of interest building as to whether or not you can target these cells because they're the same in everyone. It's such a conserved pathway. It'd be a one drug fits all kind of thing. Now, these 3 papers just all came out back to back, looking at the role of targeting MATES in TB, and I'm going to talk about our work in particular, but the work from the Glickman and Cowley labs, largely in our lab, are all largely similar here when it comes to vaccinating for mates and TB. So we started by just looking at what mates do when you in vivo, when you infect a mouse. Now we have this nice tetramer, which is the MR1 molecule loaded with 5-OPRU that John made. And so we can track the mate-specific T cells. And you can see after TB infection, they don't budge. They're just flatlined. You can see the conventional T cells that see ESAT6, which is a major TB peptide, come roaring in, but the mate's just flat. They do get activated a little bit though. You can see they upregulate molecules like PD-1 and CTLA-4. So they are being stimulated, just not enough to convince them to clonally expand. So it's kind of a weak response. If you can do this with Francisella, the mates will expand 100-fold, and here they're just kind of like, a little activation. And when we infected MR1 knockout mice, there was no difference in bacterial growth. So overall, mates make very weak responses to TB and don't have a role in host survival. But that doesn't mean if we couldn't get them to control TB. So we thought, well, what if we vaccinated for mates and had a whole bunch of mates present at the time of exposure? And you can do this because in mice, you can get mates to expand to huge levels. And that requires combining a TCR ligand and an adjuvant. And we use CpG as a classic adjuvant. You can see if you just put 5-OPRU into the lungs of a mouse, they don't really do anything, or the adjuvant alone. But if you mix the TCR ligand and the adjuvant, mates expand 50 to 100-fold. So you can see just these enormous populations of mate cells expanding after vaccination. So we did this. We vaccinated mice. We had this many mates, and then we floated a cloud of tuberculosis through the mouse cage to see if at the time of aerosol exposure you had this huge population of mates, if you could enhance control of the infection. And it didn't. In fact, there was really no difference at all in the bacterial loads if the mice had normal mates or this huge population. In fact, if you use an MR1 knockout mouse, which has no mates, the bacterial loads are the same. So really, through modulating the number of mates we had at the initial time of TB exposure, we could not impact on the outcome of TB at all. Okay, so it didn't look so great for mates in terms of a vaccine. So we were really thinking this was sort of putting the nail in the coffin of mates. But then we did one more experiment. We decided, well, let's just test it therapeutically. So we'll let the infection progress until it becomes chronic, and then start treating the mice. And the reason why we did this is because we were seeing mates were in a very different activation state in chronic TB as compared to early. So we wondered maybe if we stimulated them after the infections progress, would we help? So we treated the mice with PBS. 6FP is actually an MR1 ligand, which does not stimulate the mate TCR, so it's just a negative control, or 5-OPRU. And here we didn't even need adjuvant. The tuberculosis infection alone was a sufficient adjuvant. Just putting 5-OPRU into the lungs causes a huge expansion of mate cells. And when we looked at bacterial loads in the lungs, we got a significant decrease in the lung, but not so much in the liver. So this really surprised us, because I mentioned we simply cannot get conventional T cells to do anything like this. And in fact, I can't think of any other immunological perturbations in a mouse which will result in a log kill of bacteria in 3 weeks. So we were like, really? The cell type which doesn't do anything early is the one thing, like if you stimulate it late, you get this big kill off of TB. So we thought, well, all right, what is the mechanism here? Mates make a lot of IL-17, TNF alpha, and interferon, and a little bit of interferon gamma. So we repeated the experiment and we blocked those 3 cytokines in the context of mate stimulation. And really IL-17 had the biggest effect on suppressing the host protective effects of 5-OPRU. So we blocked IL-17, we abrogated largely the beneficial effects of 5-OPRU. So this is an IL-17 dependent protection. We don't know if it's coming from the mates because we blocked it with an antibody, but it's a fair hypothesis. So we thought this was exciting enough that we really should pursue this in the non-human primate model. And so I'm going to end with showing a little bit of unpublished data from a macaque experiment. So we tried to get as close to that mouse model as we could. We infected the monkeys with TB, waited till 6 weeks, and then started treating them with 5-OPRU into the lungs. And the first thing that, again, we got another surprise. The first surprise was that we lost 3 out of the 5 animals that were treated with 5-OPRU. Each one of these 3 animals developed rapid onset signs of distress that led the vet to make the call that this animal needs to go down for humane reasons. So it's already not looking good. we killed 3 of our monkeys with 5-PRU. That didn't happen in the PBS treated macaques. All right, so we went on, but when we went on to look at the disease progression, again, this is PET CT imaging. I'm just showing you 3 time points here, but we actually measured many. Overall, there was no difference in disease progression after 5-PRU treatments. You can see it didn't cause any reduction or acceleration of disease progression, and it was very similar between the 2 groups. So even though we lost 3 animals, it wasn't because they had this rampaging tuberculosis disease. And in fact, when we looked at the granuloma bacterial load, so again, necropsy, pull out the lung, bread loaf it, lesion by lesion, one at a time, process them all separately. What we can see is I had to plot these 3 animals that were necropsied early separately because it's not fair because they're in a completely different time point. But overall, there was no impact. I mean, maybe there, you can see I have statistical significance values here, but these are not big differences in bacterial growth. And these early increases are probably just part of the natural dynamics. You know, the bacterial loads are a little bit higher early and then come down a little bit later. So we interpret this to mean there's really not a big impact on disease progression or bacterial growth in granulomas with 5-PRUs. It didn't work. It didn't give us enhanced control of the infection like it did in mice. And there was no evidence here of extrapulmonary spread in spleen. I just want to point out that I think we know why the animals died. So one of the classic features of TB in rhesus macaques is they're prone to developing horrible lymphadenopathies. In fact, lethal lymphadenopathies. The lymph nodes in the chest can swell so big that they actually compress the airways and kill the animal. And we could see that that's what was happening upon necropsy. You open the chest and there's a gigantic lymph node crushing the windpipe. In fact, we could visualize that with CT and even quantify the amount of bronchoconstriction that happened. And you can see in each of the 3 animals that needed to be euthanized, there was a sudden onset of bronchoconstriction at the time. Now, it's already.
And did you ever treat uninfected animals with 5-PRU?
I'm going to show you that. Yeah, okay. And actually, before we did this experiment, we did nothing. They're fine, basically, but I'll show you a little bit of that data.
Okay.
Macaques are really prone to this. Joanne Flynn's published on this extensively. So we don't know if we got unlucky or if the drug did it. There is no microbiological outcome, or I'll show you there's no effect on the mates to explain this. So it's not really clear if this was bad luck or if the drug did it, but certainly we did not help TB in monkeys like we did in the mice. Now, we were worried that did we even impact on the mates? I think we very clearly did. PD-1 upregulation, again, is induced by TCR stimulation. And so we could see that after 5-PRU treatment, there was a big increase in PD-1 expression on the mates and on all of them. So they're all being stimulated, but they didn't expand. So rather than this 200-fold expansion, like we see in some of the mice, if anything, there's a trend that mate frequencies tended to decline after 5-PRU treatment. So again, this was unexpected. We thought we'd have a lung full of mates, and we didn't, even though there was clear evidence we did hit them. So we also looked at the function of the mates. And another great thing about John's tetramer that he made for monkeys, that it stains so bright, you can actually combine intracellular cytokine staining and tetramer stain. And even after TCR ligand stimulation. And so mates make, in monkeys, in mice it's different. In mice, they primarily make 17 and a little bit of gamma. In monkeys, it's the other way around. They make primarily gamma and a little bit of 17. We can see all 4 of these cytokines, even some GM-CSF from the mates. Now, over the course of the infection in the control animals, the function of the mates really didn't change. You can just see that in these FACS plots here. And in here, you can see just as this is all 4 cytokines combined, any cytokine positive, tetramer positive mate didn't really change. But in the treated animals, as soon as we started treating them, you could see just the MFI start plummeting. And then just to the point where very few mates are making cytokine at all. The function of the mates cratered after we started treating them with 5-PRU. So we didn't expand them, and we just kind of screwed them up functionally. This looks like T-cell exhaustion, but we don't want to call it that for a whole host of reasons. But there's some sort of mate dysfunction that's happening when we treat them with antigen. So instead of, again, complete opposite of mice, we had a huge explosion of highly functional mates. Here, they don't expand and they just kind of boop out. So we thought, well, this could be because of the TB infection. This could be because we gave too much of the 5-OPRU, and this is high zone tolerance, or it could be because of that PD-1 expression. So we did one experiment that would test all three. So now we did this in naive macaques, or uninfected macaques, I should say. We treated with CpG and 5-OPRU. So we vaccinated the mates with adjuvant and with 5-OPRU, and we reduced so there's no TB, and we reduced the dose 10-fold. So we've come down off the really high dose, and we did 2 groups plus and minus PD-1 blockade. And we definitely stimulated the mates and blocked PD-1 because you can see this huge increase in PD-1 expression on the tetramer positive cells after 5-OPRU inoculation. And we blocked PD-1 because we stained with the same clone that we injected in vivo to block. So we got a nice blockade. But again, mates didn't expand. So even though the animals were not infected with TB and we reduced the dose 10-fold and mates all got stimulated, they didn't increase in number, and PD-1 blockade didn't help. So this poor expansion of mates was not explained by the presence of TB, too much 5-OPRU, or PD-1. They just don't expand. And then we looked at the function of the mates. Again, we saw a real dramatic shutdown in the functional capacity of the mates after vaccination. So that's the opposite of what a vaccine should do. It shouldn't ruin the functional capacity of your cells. That's what happens here with the mates. We did partially alleviate the loss of function when we blocked PD-1. They still lost function. You can see it still goes down, it's just not quite as bad. So the PD-1 expression we're seeing maybe is perhaps in a bit modulating the function of the mates, but has nothing to do with the lack of expansion. So just to summarize this last part here, in mice, it looks very promising. If you take a chronically infected mouse, give it 5-OPRU, the mates expand to huge numbers, they're all functional, and the bacterial loads come down. We know it's all 17 dependent. In a monkey, you take a monkey that's chronically infected, do the same thing, you give it 5-OPRU, the mates don't expand, and they just poop out functionally. They enter this functionally inactivated state that looks like exhaustion. So First off, I should point out, this makes the point that we haven't actually tested our hypothesis yet because we weren't able to expand mates in a monkey. So we still don't, therapeutically, the first question, therapeutically, can we treat TB by expanding mates? Still don't know because we tried and we failed. But we got this fascinating this fascinating difference in what the mates did in vivo when we stimulated them. And this sort of poor lack of expansion and functional inactivation, we think actually, when we started reading the human literature a lot more carefully, could have been predicted, and maybe is actually the same in humans. There's a number of studies now where even with controlled human infections or live vaccinations with things like Shigella, mate cells in humans don't expand when you give bacterial infections. In all of these studies, in fact, in many of them, they start to look exhausted. So it seems like maybe even in humans, when mates encounter a bacterial infection and are stimulated by a bunch of TCR ligand, because all of these bacteria here make 5-OPRU, make riboflavin, they don't respond like a normal T cell would, like a conventional T cell would with a big clonal burst. I mean, if you look carefully, you can see alterations in the clonality. So you're getting something happening. And maybe in like this peritonitis paper, you have like a 2-fold increase in the number of mates, but that's because they all migrated in. So, but you can get these subtle changes, but there's no boom of expansion like you would do with conventional T cells. So we speculate that this is a normal response of mates. They're not supposed to undergo large clonal expansions like conventional T cells in vivo. They're there at relatively high frequencies. They're pre-positioned at mucosal surfaces, but when you stimulate them hard, if anything, they start to adapt, to downregulate their function. And we think that maybe to actually stop them from becoming immunopathogenic. But this also means that a lot of the mate field that wants to target mates for treatment of cancer or all these different infections, it might be this poor expansion of mates, or this lack of proliferative capacity, may be a big barrier to therapeutically targeting them. Because if you can't expand them, how are you going to actually get them to large populations that you probably need in vivo to do stuff? So going forward, we want to try to figure out, well, how do we get mates to expand, to go back and test our actual hypothesis is, can we target them? Or would that be a bad thing? If you make mates expand, are you going to have a big problem? I mean, these cells are chronically stimulated by your microbiota, right? They're seeing your microbiota. And so maybe you don't want lots of a cell type, but seize your microbiota. Who knows what that would do to your mucosal surfaces? Anyway, that's all just to speculate. Perhaps it's a bit provocative, but the more we think about it, the more we think that mates are just not meant to be big proliferators. Okay, so just to end, blocking, I told you 2 stories. We tried to target PD-1 to treat TB. That's a bad idea because you need this inhibitory receptor to limit bad pathology. And we have data in mice in terms of mate cells. We think it could be important to branch out to other subsets of T cells, like unconventional T cells. And there's data in mice to think that targeting mates could be really great in TB. But when we tried it in non-human primates, we encountered a very different kind of biology of mate cells. And so we think maybe if we really want to ask that question, we have to do a little bit more to understand what regulates mate function in vivo and macaques. Also makes a good point why it's important to test your mouse hypotheses in a monkey, because we've been wrong probably 50% of the time. I shouldn't say we've been wrong. The hypothesis that was generated in mice is not translated into macaques at least 50% of the time. Okay, so I'd like to end there. So the PD-1 story I told you at the beginning was, so any of these macaque experiments are on all hands on deck, so everyone jumps in and helps. But the PD-1 story I told you at the beginning was spearheaded by Keith in the lab, and all of the mate data I showed you was done by Sake, with a huge amount of help from Nikiana and Danielle, who are full-time NHP technicians in the lab. And with that, I'll stop and take any questions.
All right, thank you so much.
Thanks, Dan. That was a great talk. So should I read some of the questions in the chat? Yep, is that okay?
Sure.
Perfect. Yeah, or we have time, so we can also let people ask their own questions.
Mm-hmm. All right.
Do you guys prefer if I stop share so we can see each other, or should I leave it up?
Be nice to see each other.
All right, so I'll read a few of these questions in the chat, and then people can also, maybe if you raise your hand, then we can call on you directly as well. So from Cody Elkins, who's a PhD student now in the IMP program, could cell metabolic differences in mates explain the different responses in mice compared to macaques?
That's a cool idea. Yeah, could be. There are some big differences. I mean, there's data out there where they've compared gene expression profiles of mouse and human mates, and overall mates look like mates, but there are some big differences too. Mouse mates are all CD4 or CD8 double negative. They're double negative lymphocytes, but monkey, and they're all Th17 producing cells, but in monkeys, they're CD8 positive cells, and they look more like a Th1 like phenotype instead of toxic cell. So they look like very different classes of effector lymphocytes between mice and monkeys. Another thing is in mice, we're doing this in SPF mice. The mates all they're like baby mates, right? They're pretty clean animals. And that's not, they don't have a microbiota, of course, but they're way cleaner than a monkey would be. So we think that maybe there's also a difference in the sort of differentiation state of the mates in a mouse versus a monkey, right? The mates in a monkey are way more matured, perhaps, because they're stimulated by more stuff. And I think that's true. If you just look at PD-1, mouse mates in a naive mouse are pretty low, almost negative. But in a monkey, there's always a low level of real positive PD-1 expression. So the ongoing stimulation or the history of the mate in a mouse and a monkey could be completely different, so that the monkey mates have tuned themselves to not respond, but the mouse mates are still baby mates and have never learned to do that. But how the metabolism and all other forms of immunoregulation link into that, all great ideas at this point. We don't know.
Yeah, so I guess tying into that, you talked about sort of like the clean mice. So someone has asked, should you try dirty mice compared to the monkeys?
Yeah, we tried. Our vets won't let us get anywhere near that, but I hope someone does that.
Yeah. So Rafik had another question back to, I think, related to the PD-1. So TGF-beta blocks anti-PD-1 rescue of T cell function. So did you look at TGF-beta in the tissues of infected non-human primates?
We didn't. We should go back and look. Yeah.
So yes, I see John. We'll come back to John in a second. Maybe the last question from Rabin is, across your parallel mouse non-human primate experiments, are lab mice too skewed toward T cell function and thereby poor predictors of pathology in larger mammals that may be more myeloid driven?
That's an interesting question. I don't know if I would phrase it like that. because we know in humans even, you need, without CD4 T cells, you are enormously susceptible to MTB infection. There is, however, I would put it like this: the quality of the T cell response is very different. So we've spent a lot of time agonizing over the flavors of CD4 T cells you get responding to mice and versus humans and monkeys, and they're very different. And the short of it is that the mouse CD4 T cell response is way too Th1. It's very heavily focused. It's a pure Th1 response, even generating a lot of terminally differentiated or highly polarized Th1 cells. And you just don't see that in humans and in macaques. It's way more diverse and not so heavily polarized towards Th1 phenotype. It's more of a, we call it Th1 star, because it's got with a mixed properties of Th1 and Th17 cells. So one of the key differences, I think, has to do with the quality of effector CD4 T cell polarization. I wouldn't say it's a macrophage CD4 thing. I think you need all of it in all species. I don't know if that was clear.
Yeah, so no, okay, so Vijay had asked, I think, a follow-up question about the activation of the mate cells, and so they could express CXCR5, making them go to either lymph nodes and germinal centers. So did you look at that?
We did. We actually looked in many tissues. I didn't show it for the sake of time. Mates did not expand anywhere. We looked in 10 tissues. We could identify mates everywhere. They didn't expand anywhere. They become activated everywhere. It's interesting. You put 5-HT into the lungs, and they get activated in all tissue. So there's a systemic activation. Even though that ligand is really short-lived, when you get it into a monkey or into a mouse, by the way, you stimulate mates everywhere. But they did not expand. So it's not a redistribution effect. They simply do not grow. That was a good question. Thank you for asking that.
Yeah, so John, do you want to go ahead and ask?
So, yeah, first of all, for the benefit of everybody else, and Dan, you've already hinted at this, referring to mouse mates as baby mates. People, each of us in our blood, 1 to 10% of our T cells are mates. So we've already got plenty of mates around in circulation. I don't know, what. Anyway, so there's quite a bit of difference between human mates and mouse mates at the basal level. The question I asked in the chat was, do the human or non-human primate make granulysin? And so are there other effector molecules? And granulysin is present in humans, I think in non-human primates too, but not in mice.
I should know that. That's a great question, John. I should know that. I know their functional profile, again, is very different. Again, mouse mates are very Th17-y. Monkey mates are way more Th1-y or TC1-y and have more cytotoxic molecules. But overall, mates, in TB, they, like, for example, I don't know about granulysin, but granzyme B, very little granzyme B expression by mates at baseline. And after TB infection, maybe a blip if you stare at it long enough, you'll believe it, but never super statistically significant. So it's nothing like, you hear a lot about mate cytotoxicity based on stuff that you can see in vitro, but you look at a polarized killer CD8, conventional CD8, and they got 2 logs of granzyme B, and mate, it's a nubbin that comes up out of negative. So I'm not sure cytotoxicity is really their jam. I think in vivo mates are cytokine producers.
Do they score in CD107 assays?
Even CD4 T cells score in CD107 assays. So it's,
Okay.
it depends on MFI cutoff, right?
Yeah.
That's a tough assay. So yes, but yes and no. There are, don't get me wrong, you can find a lot of granzyme A, but you can hardly stain for perforin. And I think we even found granzyme K once by flow, but you see all those in CD4 T cells too that you wouldn't necessarily think of as a bona fide killer. But again, that's I'm trying to be very honest in my opinion. I just don't know that we have good evidence that cytotoxicity is a major function of mates. Then again, who knows? With all this data about viruses in mates, it could be.
Okay, thanks.
Are there more? Just looking to see if anyone else had their hand raised. Anyone else want to ask any more questions?
I have a question, Dan.
Hey, Dan.
How are you?
Good, man. How are you?
Do you think antibiotic therapy would affect mate activation or the set point of it?
That's a cool question. I hadn't thought of before. we're actually looking, this is not your question, but we are running the microbiota now of the monkeys that were treated with a 5-OPRU to ask if we might have impacted on the microbiota by stimulating the mates. But I've never looked myself as to if TB chemotherapy, for example, may be having an impact on mate function because of the kill off of the microbiota. That's a cool question. Someone must know that, just not me. Again, I'm not really a mate guy.
Neat way of thinking about them, though.
Thanks, Dan.
And yeah.
Yeah, thanks, Dan. So I was very interested to hear all of this work, but also your hypotheses and speculations. And so one of the things that struck me is when you said you have these populations of immune cells, and this is, of course, must happen a lot and in a broader context, that these cells are here to protect us against pathogens, but at the same time, there is this fine balance with not protecting the beneficial microbiome. So you mentioned a little bit about this as a potential explanation for the populations of mate cells that are there. What is there evidence for their role in maintaining the microbiome or not attacking it?
There is in mice, mate deficient mice have altered microbiomes, but I'm not so sure if we know it's a beneficial or detrimental microbiome. Again, the role of mates is still a bit mysterious. There was one report, I thought was a very important paper, where they identified a single MR1 MR1 deficient human. And they had an interesting history of infections, nothing life-threatening. They had, I think, a severe intestinal Campylobacter infection, and then they had a severe lung VZV infection, and then they got older and I think they left scarring, and then they got older and had a tattoo and got really bad warts from the tattoo. So, you know, in the first MR1 deficient human that was described, you found all three barrier sites got infections, and 2 of them were viruses.
Okay.
I don't, you know, in all of the new data about COVID and mates being really associated with outcome, who knows, they could be playing unexpected roles too, even though see the microbiota, maybe they're somehow modulating viral infection. So they could be doing something very surprising.
Very, very interesting. So it is 1 o'clock. So let's thank Dan. I'm not sure about the schedule, but if there is a few more questions, please hang around for another few minutes. But everyone who has to go, please feel free to do so. And let's thank Dan once more for a fascinating talk. Thank you, Dan.
Thanks, everyone.
Thanks, Dan. Okay, so I think I'm talking